Hello and welcome everyone to the 7th meeting of Genelia's Scientific Visualization Interest Group. This talk is being recorded and will likely be published on our YouTube channel. I'm including the link here on the chat. So next meeting, we're going to be hosting John Stone from the University of Illinois at Urbana-Champaign. He'll be talking about real-time ray tracing in VMD, his molecular visualization tool, and other aspects of using GPU acceleration for scientific visualization and simulation. Should be a pretty cool talk with lots of nice videos. But today I have the pleasure of welcoming Robert Krueger. He's a postdoctoral research fellow at Harvard's Visual Computing Group, and he's also a senior scientist at the Laboratory of Systems Pharmacology at Harvard Med School. His research interests are in the field of data visualization and visual analytics, particularly spatial and spatially referenced multivariate data. So pretty much the kind of data that we have in abundance here at Genelia. And so with that, I'll turn it over to Robert.
Thank you very much for the introduction. Let me share my screen. Just as a quick check, does everybody see my slides?
Looks good.
Looks good. Okay, thank you. Yeah, so hello, everybody. And I am feeling very honored and I'm very thankful to have the opportunity to speak in this group today. As Bill already introduced, I'm Robert Krueger, and my talk today will cover a path from visual analytics to interactive storytelling for large multiplex cancer images in the field of digital histopathology. So before I go into more detail, I would like to introduce me and my working environment a little bit. So I'm a visualization researcher and I did my PhD in Stuttgart, where I worked on geospatial visualization. And now I'm a postdoc at the Visual Computing Group, led by Hans-Peter Pfister, and also a senior scientist at the Harvard Medical School at the Laboratory of Systems Pharmacology, led by Professor Sorger. And this setting has been very fruitful in different ways. So at the HMS, we have a lot of people, biologists, pathologists, working on biomedical research problems. And what we do with the Visual Computing Group is we interview them, we derive higher level domain goals from them and specific tasks they wanna get solved. And then we basically do a design study usually. That means like we interview them and from the task we suggest different like lower level features that the tool should have. And we apply and implement these features in prototypes that we then evaluate together with the biomedical experts. I haven't been working on this alone, so I have a subgroup that I lead, and I just want to highlight 3 people specifically that also contributed a lot to the tools and approaches you will see today. And this is Salman Balchol, Jared Jessup, and Naan Huyn. So yeah, now I work specifically in the field of histopathology, and the field of histopathology studies manifestations of disease, including cancer. So historically, by direct visual examination through a microscope. However, with digital histopathology, a rapid shift towards digital instruments allowing for computational analysis is now possible. And this is something referred to often as scope to screen, from microscope to computer screen. And with this shift, new imaging methods are developed, of course, that produce large and multivariate data sets. For example, CISIF, which stands for cyclic immunofluorescence imaging. This is a method that was developed at Harvard. And a lot of the data sets and the tools that you will see today work with this data. And so I want to take a minute or two to introduce this imaging technology and the data structures that we're working with. So yeah, as already said, CISIF stands for immunofluorescence imaging and was developed at the LSP at Harvard. And now I want to quickly run through this imaging process. So as a first step, antibodies are put on a tissue, currently limited to 4 antibodies at a time, but there are like newer microscopes that we already experienced that can do like 20 to 30 in one shot. These antibodies are conjugated with different fluorescence dyes, making the molecular state of each cell visible. The tissue is then imaged and digitized, yielding images encoding the location and the amount of various proteins at the level of individual cells. And then subsequently, the labels are chemically removed and the process is repeated with labels for different proteins. And this cyclic process is really key to measuring a large number of molecular features, which offer great insight into normal and also disease processes in cells. Overall, this results in up to 60 images, each highlighting different reactions of cells to these antibodies and thereby revealing their function. And this resulting data is really very big, often up to 100 gigabyte, and images, and the resolution is often like up to 60,000 times 60,000 pixels in size, containing more than 1 million cells. So now the data is processed. That means registration, and I want to highlight this. It looks, but it's not 3D data, right? So we have these different images from the different cycles, but still we need to register them. And sometimes also we have multiple images that have to be stitched because they were separate and now they want to be combined to the original tissue again. So stitching and registration. And then after that, segmentation of the image data into single cells. Here we use a UNet, a convolutional network segmentation process. After the segmentation, then the segmentation mask is used to extract per cell information into a structured data table where each row represents a cell and each column encodes a reaction in an image channel. So basically for every cell that contains, of course, multiple pixels, we do a mean value of the intensity value that usually ranges between 0 and 2 to the power of 16 for 16-bit depth. So we have an intensity value for each cell for each channel. So to summarize, we end up with 3 data sets: the image stack, the segmentation mask, and the single cell feature data table. Now, on the one hand, the digital data sets allow us to apply, for example, machine learning and other computational approaches to reveal patterns and classify cells really at scale. But then, on the other hand, manual analysis is also needed to leverage the hard-won information that is known primarily to the pathologists. And the pathology process includes, for example, panning and zooming across specimen, switching level of magnification, measuring regions of interest, and even dotting regions of interest with a pen on the tissue when we look historically how they used to do things. Now, of course, with digital histopathology, we have new opportunities, and the question is really like, how can we adapt these practices to a digital environment and to semi-automated analysis, combining the interactive visual things with machine learning. And a suitable methods that draw on human capabilities to rapidly process this information and to detect the patterns by eye is visualization. And often visualization is primarily used as a means to present or communicate the results after data preprocessing, after the analysis. And that is, of course, very useful and helpful to do. But visualization can additionally be also used as a means for quality control throughout the process, as a means to improve the pre-processing and to enable interactive analysis with the human expert in the loop. And that's really what we're aiming for here, to not only use visualization as a means for presentation, but throughout the analysis. And today I want to show you 5 of our applications that address different challenges along the analysis pipeline. And that ranges from, as you see here, pre-processing on the left, where we have tools to kind of clean the data, especially the resulting feature data in this data table. We have prototypes for region of interest exploration. So this mimics more like the traditional pathology exploratory process, where you can magnify and measure regions and annotate things. We have work on cell typing. So how can a user be supported in defining what cells are immune cells, cancer cells, and so on, by looking at the tissue and by using machine learning. Then we step forward into cell-cell interaction. That means we're not looking at cells individually, but we're looking at more like what other cells are in the neighborhood. Are there any spatial patterns that these cells form to better understand the cancer microenvironment? And then lastly, we will look into storytelling. So after all that is done, like how can you communicate this information, or how can biologists create such interactive data stories that we can then share on the web? Okay, before I go into detail about these tools, I just want to quickly talk a little bit about our visualization strategy here. So of course, the data is really big. That means like we have 100 gigabyte. So every channel could still have like, couples of gigabytes. And of course, when we do web-based visualization, we don't want to load that at once into the browser. So as probably familiar to people here, we use a multi-resolution approach where we kind of pre-aggregate the data into an image pyramid. And so when the user pans, we really just need to load respective tiles of the current zoom level. When the user has fully zoomed out, we can just deliver an aggregated version of the image to the client, because we don't need all the detail. And when the user zooms in, we can just deliver the certain part in the viewport to the client. So we never have to deliver the whole data at once to the client. Now we have multi-channel data. That means we have such a pyramid for each of the channels. And we can deliver the tiles from these respective pyramids to the client. And then we do the rendering on the client side with an adaptive color model. So in that case here, for example, we get this magenta color when we have the blue and red channel here for two channels of choice activated. We can see how that looks in the tool. And this is like a basic feature that most of the tools I will show use. So you can mix these colors here together. Usually good to choose well-separated colors, here in this case RGB, and then you can basically, they're combined in the image space. We do the same thing for the segmentation mask. So you can see like we can use the segmentation mask to allow the user to select individual cells on an individual basis, and then also to use a lasso tool to draw a region of interest. And then, of course, with knowing the IDs from that segmentation mask, we can link that to the tabular data. Okay, so the first tool I want to focus on today is for pre-processing, and especially in that domain, they call this approach, what they do here, gating. And I want to elaborate a little bit on what that means before I show the tool. So as we remember from the pipeline I showed, the segmentation mask is used to extract information into the structured data table, right? And that structured data table is then used a lot also for all kinds of clustering and downstream analysis these experts do. So of course they have an interest that this data table is well, the data is cleaned and there are no outliers in it and everything is kind of, filtered and normalized correctly. And but often the image quality is not optimal because this is a very experimental setting. You try out new approaches, you try out new antibodies, you try out new machines. Different people work on it, of course, in a research setting. And so when we look, we have to see like when is actually a marker expressed in a cell, and that can vary a little bit between data sets, of course. So when we look here in this region and I marked a few cells, we could say, okay, for these cells, the marker is well expressed, it clearly lights up. But then there are some other regions in the image where that might be not as clearly the case. And now the question is what to do. Also, sometimes we have something called like a spillover where the marker basically is light up in one cell and it spills over a little bit to the next cell, even that cell doesn't really express that marker. And to counter that, basically, we want to support a process called gating, where we basically threshold what ends up in this data table depending on the marker expression levels. So this is a data table and just pretty simple, like for one channel here for CD3, we could basically set a filter and say, like, we only want to consider intensity values in the image that are above a certain level and below a certain other level. Because when it's lower, then we don't really consider that to be expressed. And when it's higher, it could so for example, depending on the data set means that there's just like, it wasn't, the bleaching didn't work well, or there's a certain artifact or so. So when we apply that, we could basically set cells where we think the value is not expressed to 0. And of course, that's really hard to do without really looking at the data. And so we build a tool. And does it mean like we have to do that all manual? No, we don't have to do it completely manual, because when we look at the intensity value of the image, they are very skewed, as you see here on the left. But when we do a log transformation, we get nicer curves. And it turns out we can apply a Gaussian mixture model here that kind of nicely separates these intensity distributions into 3 very nicely distinct Gaussian curves here. And we can see the left one, the blue one here, is usually referring to the background in the image channel. The orange one here, the middle one, is usually well capturing the signal between the cells. And then there's one curve here on the right, the green one, that is usually capturing the real expression signal of the cells. So using that Gaussian mixture model helps us a little bit on the color mapping and the transfer function usage. So this is the tool to do this gating process. Here on the left side, we have, as you saw already earlier, the channel selection for the image data. In the middle, we have then the display of the image data. And here on the right side, we have kind of the same thing again, but this is not for the image data. This is for the table data. So here you can filter what's in the tabular data. And then depending on what we set here, we have an outcome of the data, of the cleaned data set, where certain values are, for example, excluded. Now, here's a video. Hopefully this is fluent on your end. So in this case, we activate the first channel here, CD45, an immune marker channel. And we can color that, of course. Set that to red here. And then we can click on auto, and it automatically tries to get this third Gaussian curve. So this is nice for a quick first fitting of the transfer function here. And we can do that for multiple channels. And then we can look at the tabular data. And by activating that, we get the segmentation mask. And we can change here the gate. And everything that is still shown here with the segmentation means will end up in the data table. So by changing these values, we can set the thresholds, the min and max threshold, for the filtering of this final data table. We have an end mode. That means we always, when we look at multiple channels for the end mode, we see a surrounding of the cell with the segmentation mask here when it's still considered. And we have, was a little bit too fast, we have an OR mode that basically shows individual channels and how they're filtered.
Got a question.
Uh-huh.
So when one of your analysts or scientists goes through this process and comes up with a nice bunch of parameters,
yeah,
is there some way to spit that out into a script and automate it?
This is what you see right here. This is basically, you can download that for the CSV gating and also for the channels, and then you can apply it to other data sets. Often our users have cohort data. That means they have, you know, I don't know, 20 of the same experiment, kind of different from different tissue, but same markers applied and so, and they just want to do it on one data set. And then as you see here, they can just load it, and that is applied to the next data set as well. Is that?
And if they want to do it on a cluster to a whole bunch of data sets all at once?
So basically, we don't really, so when they want to do it all at once to a bunch, they would execute, I guess, this thing computational without their interface, I would say. We haven't really provided that code piece, but it is basically possible because, like, this is a client server system, and the filtering code, I think, is very easy to extract.
Okay, thank you.
Mm-hmm. So yeah, this is like, you know, not rocket science, but a really effective tool. And we have like more and more users using that for their data cleansing, because for them, it's really important to have a visual feedback of what they do here. The next approach I want to look into with you is region of interest exploration. This is more addressing the typical a process, exploratory process, a pathologist would usually do manual by looking through a microscope, and we try to mimic that and bring that more to the digital environment. So as you can see here already, this is a focus and context approach with an interactive lens that you might be familiar from other domains. Basically, the idea is that you can focus on a region of interest and still see the surrounding context. And we work together with pathologists from the Brigham and Women Hospital that we have had meetings over like several months to understand their tasks. And we developed this tool iteratively with evaluating the interface with them. So in the end, we got a very nice workflow here. And as a first step of this workflow, the tool provides scalable and interactive image viewer that you already saw to select the channel and do the color mapping and to explore the data from overview to detail in the image viewer. I don't need to show that because it's basically similar to all our tools. But subsequently then, when a user finds a region of interest, you can use a lensing technique, focus and context-based lensing technique for close-up inspection of the image and feature data. And here we basically implement a digital lens where we draw on the familiar experience of pathologists to inspect the data through an eyepiece. And the lens can be adapted in shape and in size. And as you can see, it has a measurement. You can zoom in. When you zoom in, we basically grab different layers of the image pyramid so that we can still maintain the optimal resolution. And then in addition to these easy functionalities, we have additional features for this lens. First of all, we have image-based features. So here you can, for example, see while 3 channels are active or 2, the user can use this lens to isolate a channel and to look at specifics, or the user can have a certain setting of immune markers within the lens while having a more immune cancer a tumor structure outside of the lens to look for immune-specific reactions in context. We also have a split screen lens where the user in this case can compare SciSIF data to adjacent H&E data. So looking at the morphology and the anatomy of the tissue at the same time and comparing things. And then we also provide some more like single cell statistic based features where we link through the segmentation mask and the information about cell IDs that we have, we link the image data to the extracted feature data and additional information in that feature data. So here we basically see histograms for all the active channels. In orange is the whole data, in white is the respective part in the lens. And while we hover over, we can basically look at these distributions. We also have a more compact representation where you can see the different distributions for all the markers. And you can see basically the orange would be the overall data set, and you can see these white peaks, which indicate either more or less in the area of focus. This one here shows results from cell typing, where we show the outlines based on the respective cell type. And so there are different features that link the image and the feature data and allow the users to explore regions of interest. Then, as the data is very big, this can of course be a little bit time consuming. So we provide certain means that help the user to find similar regions. In this case here, the user can set a threshold, and then the user can click somewhere and we visualize a heat map that detects spatially similar regions in the image. This works at the global scale, but also at the local scale. When we zoom in at these mucosal structures here, we can see that the approach is able to capture these things, and we can switch from this kind of heat map approach to outlines. I don't have a slide in this talk here that goes into more detail, but it is a histogram-based approach. So what we basically do is we compute into intensity histograms for the region under the lens, and then we have a sliding window that goes over the image space, and we compare what's in the lens to the sliding window with a fast histogram propagation approach that makes it interactive. And we can do this for one channel or channels in combination. So then lastly, when the user finds something interesting, she or he can annotate that along the way. As you can see here, The user can take these kind of snapshots when something interesting pops up. The user can make these snapshots, and that all is shown here on the right side in this dotted panel, where the user can give this annotation a name and a description and can save that to the database for later usage. And you also see on the top we have a search bar where you can then, let's assume you have hundreds of annotation, you can search by keywords and descriptions. And you can also click on them and it will navigate you back where the annotation appeared. And when you click on that, we restore the lens with all the settings it has to go back in the exploratory mode. This is Scope to Screen. We published that last year at IEEE Viz. I don't know, like just quickly checking in if there's some specific questions on that approach. Otherwise, I would continue to more machine learning based next approach for cell typing.
So what is the software stack?
The software stack, sorry, what do you mean?
How did you build it? What is the underlying libraries?
Oh, I see. Okay, yeah. I could have a very good question. I should have provided more details on that. So the software stack it's a client-server application, a web-based application. The server side is Python Flask, has a RESTful API, and then the client side is JavaScript. For the rendering, we use OpenSeaDragon. That is like a client-side JavaScript library for panning and browsing large image data in like an intelligent tile-based way. And we enhance that with WebGL rendering technologies.
What was that called again?
OpenSeaDragon. OpenSeaDragon, yeah.
SeaDragon, okay.
Yeah, and we enhance that with additional technologies to render the segmentation mask and the image channels with WebGL, which is like a little bit faster when you want to do, pseudo coloring on the client side in a fast way. Yeah.
Thank you.
Yeah, sure. Maybe a little bit what I can also say, like, querying the tabular data, that is also living all on the server side. And we have a, index structure there. So the user basically, with the lens, we do a range query that is a query to the API on the server side. There we use the index structure to get the cell IDs and the features we want, and then deliver that chunk back to the client. So then another approach we're working on focuses more on cell typing. This is not necessarily in order. So this is an older work from 2019, where we focused on cell typing. So you can imagine you have all these markers, but then you want to know, is this a cancer cell, a tumor cell, and so on. So cell typing is really an essential task and is done based on these expression levels in the respective image channels. And first level cell types are, for example, tumor, immune, and stromal cells, as you see here on the right side in this hierarchy. But of course, these can then split up into further subtypes of certain immune cells and so on. So you can imagine like a really large hierarchy of cell typing, right? And a challenge is to do this at scale for not a few, but 1 million cells, while still keeping the expert in the loop. And with our tool Faceto, we draw on a combination of unsupervised and supervised learning and multiple linked views to do that. So this is Faceto. And in the center here, you see again the already familiar image viewer with similar functionality than Scope to Screen. But now this viewer is surrounded with additional feature views. In Scope to Screen, we basically had more the idea of having all the information nearby the lens, nearby the region of focus. But this is a little bit more complex feature data that we show here. So we chose a little bit of a different design approach and have these multiple linked views where we have more space for the feature data. So these feature exploration views comprise, for example, as you see here at the bottom, a table, interactive data table. That we have a direct view of the single cell data that we have extracted, and then also some projections and so on. And then what I want to focus on first is this hierarchical faceting view that is used to facet the data into meaningful regions and cell types. So as I said, this interface provides a tree view that allows to hierarchically subset the data into meaningful regions. In this case, the data is manually faceted into spatial regions. This can be done in a hierarchical fashion, and then each subset is represented in the faceting view and can be selected and further divided and exported. So here we see a little video. In this video here, the user marks a certain area and then can say, I want to create a new subset out of that. And you get this new node here that also shows a fill stand of like how many of the original cells are still in this filter. And then you can hierarchically go on and continue to divide this data set into certain regions. And that is also stored. So we store all the IDs of these regions. So later you can click on that and you get that subset again. Okay. Now you can imagine that that is quite okay for larger regions, but of course it doesn't scale to cell typing individual cells. You would sit there forever. And this is where we leverage machine learning. So how do we do this? Well, we support machine learning in two ways. First, during bottom-up analysis, unsupervised learning can assist the user to detect groups of similar cells. More precisely, we use an expectation maximization algorithm that is a good compromise for an interactive system, as the algorithm is fast, but also detects clusters of different size, density, and shape, as opposed to, for example, k-means or more simpler algorithms. I want to show you how that works. So the user can select certain channels that should be considered for the clustering and select a region of interest, and then select a number of clusters for the clustering and can execute that clustering. And then the clustering is executed and automatically provides colors for each cluster. And you see how that looks then in the image space. And now you can use the faceting tool to extract these clusters into meaningful subsets that you can then store for further analysis, for example. You can also give them a name. And so hierarchically, you can cluster your way through the data set and define groups of different levels. So now we can activate this one, and you see we only see this the cells in that specific subset. Okay. Then when a user has explored the data for a while and has gained enough knowledge, she or he may want to apply it to other image regions and data sets. And this is where supervised learning comes into play. Users can train a classifier on the clustering results or also on manually labeled data, or both. Precisely, we use a convolutional neural network here directly on the image tiles as input to classify cell phenotypes. We can see how that works here. So for a region, the user can say you want to train a classifier. You can give it a name, and then the algorithm runs. This is a small region. Depending on like how big it is, it can run for like 10, 20 minutes or whatever, like depending on their data set. But then once the classifier is trained, you get a confusion matrix that tells you how good it did. This is just the fast one, so we got 84. It's not bad, but when with experts we achieve higher accuracy levels. But anyways, when you have that, then you can apply that to other regions. So for another region, you can select from a list of pre-trained classifiers and then apply it to that other region. And then you can classify that other region in the image accordingly. So that was all about cell typing. But cell typing doesn't necessarily give you all the information about the cancer microenvironment and how spatial patterns form that could be very important to understand certain reactions in cancer tissue. And that is why our most recent approach focuses on cell-cell interactions and a spatial neighborhood analysis. So here's an example on the left side of a melanoma tissue, and then zoomed in into a certain area. And what you see here is like this red and white parts and green. These are immune cells that kind of surrounds the tumor. And when we zoom in, we can see that some of these immune cells, the FOXP3 detected ones, are able to infiltrate in the tumor, while others, the CD8 cytotoxic T cells here not that much. And this is, for example, just one of many patterns that biologists are interested in studying and understanding. So basically, it's really not only about cell typing, but in what surrounding certain cell types appear. And there are, of course, multiple ways to look at that. There are image-based approaches, of course. There are graph-based approaches, drawing like on motif discovery and things like that. And we have also looked into these, but we found for our data sets and as a first shot about the whole slide image, we go with a distance-based approach using the extracted tabular data. This is mostly because we found the motif analysis algorithms not very scalable for these large amounts of cells, like often 1 million, with also additional, like, cell types of like 10 to 20 different types or so in an interactive setting as we usually work with. So our approach here is distance-based. So in order to compute the neighborhood, we do range queries for every cell. We look like what other cells, and this is already type data, so every cell has a certain type, not looking at the original marker values. So for every cell, we look what's in the surrounding neighborhood. And then based on that, we can compute feature vectors. So we count the number of blue cells, red cells, and brown cells here in this case. And we also do an inverse distance weighting. So cells that are closer get a higher weight than cells that are further away. With these neighborhood vectors, then we can build, we have a matrix basically of number of cells and number of cell types. So for every cell, we have this neighborhood vector that describes the cells. And then we can, of course, work with these neighborhood vectors instead of the markers itself. So we can say we cluster these cells based on their neighborhood, for example, right? And in order to identify if these patterns that we found out are significant, we do permutation testing, because it could also be that these patterns occur randomly in the data, right? And so what we do is we mix the data, we mix the cell types, the positioning in the data multiple times, and compute patterns in order to get a p-value and see how significant the patterns in the original data are compared to the randomly distributed ones. So this is the overall computational pipeline on a higher level, how we quantify neighborhoods. Now let's look how we apply that. So after the neighborhood quantification, we can first get an overview of our data. In this case, we worked with cohort data sets that often have, there are multiple specimens within one of these data sets. So we build a small multiple visualization at first, where the user gets a general overview of the different data. And you can see here, the user can zoom here, but you can also click at one, and then you get the whole tool focusing on that one specimen. And of course, we can use the already introduced features of channel visualization and so as well. Now, again, focusing more on exploratory means, the user can explore this data. In this case here, for example, we can zoom in, and then we can mark a certain region. And here on the right side, we get statistics for that area. So let's assume this is the area. Then on the top here, we have a parallel coordinate plot that tells us for that area, basically the composition of cell types for that area. So we can see this area is very rich in B cells and CD4 T cells, for example. Then in the center, we have a neighborhood embedding. This embedding is a little bit different than a normal embedding, because usually points in the embedding that are close means cells that have similar markers. But in our case, if points are close in the embedding space, that means they have a similar neighborhood. So when we zoom in into this embedding, we can mark a certain region. And then we get all the areas in the image space that have that similar neighborhood. So I can show it again. So we mark a certain region, and then we get all the cells in the image data that have that similar neighborhood. And you can already see like spatial patterns appearing here. And then lastly, we have a pairwise correlation where we looked, there was a specific request from the people we work with at a pairwise interaction of cells and how often they appear near each other. Then bottom-up analysis can, of course, again be supported with automated means. In this case here, we can execute a clustering, and then here we can step through the clustering, and that detects different patterns of similar neighborhoods, as you see here. And that goes a little bit fast, but this is tonsil data set in this case, and we basically, one of the earlier patterns was all the B cells. We have detected germinal centers. There was a cluster. Maybe I can replay the video. So I'm not a biologist, of course, but I was told, that there's a mix of like germinal centers in there where, which are areas where B cells, a type of white blood cell that proliferates before migrating out of the tonsil to secrete antigen binding antibodies. Then there are epithelial cells. We found clusters of numerous T cells of various types and so on. So we basically verified that our approach is capable to capture these patterns in of known biology. Then we also have top-down analysis. So in this case here, it's the wrong video, I guess. So the user can, for example, say I have a region of interest, and now I'm curious if I can find similar areas of interest. So you can execute a search that finds similar regions within the tissue. Another way to do it is you can sketch a certain composition. So you can say, I'm interested in, certain T cells and certain B cells that interact with each other. You can sketch that in the neighborhood composition, and then the tool highlights all these areas in the image space. Okay. Then finally, you have extracted these patterns that you were interested in, and then we provide ways to compare them. So you can go back to the cohort view and see where these patterns occur in the different specimen of the tissue. And this can also be extended into a grid view where you can then compare among different specimens. Okay, so this is all about cell-cell interactions. This is like one of our latest works. And then I just want to quickly, and I know the time is already proceeding, but quickly jump into storytelling. So this is one of our maybe most well-known tools, as it is used as a tools also for public outreach, Minerva for interactive storytelling that is mostly driven by my colleague John Hoffer. And this really splits up into two tools, one tool for the authoring of these interactive data stories and one tool for displaying them on the web and consuming these data stories. The first one, the authoring, you can basically upload, your data, and then it gives you different form fields to create this interactive data story. So first of all, you can again choose, the channels you want to use for these data stories and give them names. You can choose the colors, but then you can also choose different annotation types. You can write little information of what people see here. You can add annotations and arrows. And you can also add a certain amount of interactive data plots that you want to provide for your story. So in this case here, I believe we add a scatter plot. So you can talk a little bit about what the scatter plot shows. You can select that type of visualization. You can define what's shown on the axis and so on. You get the idea. And then later, this is Minerva story that then displays this interactive data stories. And you can step through these waypoints that you created here. And you also have a audio narration that you can play. And it's really, really nice to showcase your research to others and guide people through these data sets and these findings. Yeah, just maybe quickly as a future area of research, I think there are three types that I'm personally very interested in. One is combining image and spatial omics data. So it's like there are more and more scanners that can record both imaging data and additional spatially referenced RNA sequence data, sometimes for areas, but newer ones even on a single cell level. And that means we not only have the image data, but we have for every cell spatially referenced feature vectors, high-dimensional feature vectors. And it will be very interesting and challenging to think about how can that be displayed in combination. Then secondly, looking at these interactions, so far we do that distance-based, right? We assume that when things are close, that it's more likely that an interaction happens. But I think it could be interesting in the future to look really at the marker distribution, right? When we also have higher resolution, we can look at the marker distribution and see if marker polarize somehow, either like, two cells that are close to each other, the marker goes in like the direction where they interact, or the marker shows that they repel each other. And we can look at that and define interaction more on the marker distribution. And then certainly 2D to 3D. Of course, with 2D, we only have one slice through the tissue, and we would probably lose a lot of information. And so I think looking at more 3D data is really the future. And that goes, of course, from multiple channels to multiple volumes, and that brings along very interesting question of how to visualize multi-volumetric data sets in a way that you can also interact with it. How do you segment in 3D? How do you explore things in 3D? And how do you extract features? And with that, I want to say thank you. And I want to acknowledge all the people I work with, especially Peter Sorger, Sandra Santagata, and Hans-Peter Pfister, and all my colleagues at the VCG and LSP that contributed greatly to this work and made that all possible. Thank you.
Thanks very much, Robert. It's very impressive.
I have sort of a question about usability. I work with a lot of systems biologists, and are you acquainted with Cytoscape? Have you ever seen Cytoscape?
Yeah, I know Cytoscape. Yeah, the graph visualization tool, right?
Yeah, they've spent a lot of time doing stuff with Cytoscape, and they find that they can make really nice diagrams, but then when they want to make a similar diagram, they can't remember how they did it.
I see.
And so what they've been doing, what some of them have done, not all of them, but some of them have done, is they move over instead and use something like a Jupyter Notebook-based approach, where they're actually writing basically code to derive their visualizations. And then when they want to do something similar, they can go back and they have it all documented.
Yeah.
Can you drive the kind of thing that you're talking about in that matter, where you're basically building something that if you want to go back and make it into a subroutine, you can do it? That kind of stuff?
That's a super interesting topic. We can partly, but I think there's also a lot of additional opportunities there. So I have two examples in mind. When you remember the scope to screen approach that I showed and the annotations, right? So these annotations we call them so-called rich annotations, because we don't only take an image, but we store, the area where the region of interest lies. We crop out a mini data sets from the CSV file, and we store the channel combinations that were used to derive that. And that is stored. So you can basically restore that by entering that information. The tool can go back to that position. I think that goes a little bit in that direction. And then for the Minerva story tool that I showed at last, you have a URL that defines basically on which waypoint you're on, which annotations are set on the image space, and the zoom level and viewport. So when you can share that URL, and it will bring other people directly to that position. But I think that can be extended greatly in terms of analysis results, so that you have you kind of capture a state that is described in a structured way that you can re-enter in the tool, either programmatically or by loading it through the interface. I think that's a very powerful idea.
Okay, thank you. Very nice talk.
Thanks.
Any other questions? So I guess I'll ask a couple here. So going back to the, sort of the ability to change the pseudo coloring of your visualizations back to, I think it's maybe scope to screen where I'm just wondering about research in terms of manual versus sort of auto-optimized ways of representing the color scheme and the various other things, because I know that you were showing, for example, you could go to the left, you could, manipulate the RGB for any particular class, that you had the Gaussian mixture models and all that other stuff. How much research has been done in terms of finding sort of optimal ways of mapping, but also from the perspective of having a general idea of the importance of all the different channels?
Yeah, so there are endless antibodies that people use. So I think there's like an extensive list of antibodies, and then there's a sub list of that people use more regular, right? And so I think in terms of like which channels are important, it really depends on the use case and on the experiment. It also is that a certain combination of channels often has a meaning in terms of phenotyping or so. So there's a hierarchy, and there's definitely like channels that can be grouped together. They all show immune, they all indicate immune cells or something like that. But it's really, I think, depending on the type of cancer and the experiment you look at. I think in terms of like perception and color, there's a lot still to do. Like, for example, using perceptually aware color schemes, that compensate the nonlinear perception of color that we as a human have. And I think like this is something that is very important that we look more into. I'm also interested, like, hearing from others some suggestions there, of course.
Because just what you were saying there, that if there's some sort of model about how you can compensate for the way that humans actually might have a very, that as you traverse that space, that their perception is quite different, you could actually put that into the sliders themselves, right? So that, like, as you're altering or moving through that space, you don't get into areas where it's obviously not optimal.
We should
or
use,
maybe compensating
another
for
like
color blindness, right?
Other color models that are compensating these perceptual issues. That's a good point. I think for now, like, at least what's important, is like assuming you have 3 channels on, right, and with an adaptive color model, that you choose, colors that are well separated in the beginning, at least, like RGB, right? Because when you choose like colors that are close to each other, also it's really hard because they mix in the image space. It's really hard to understand where that color comes from, right? It's important to have them well separated. And I also think it's limited to the amounts of channels you can show at the same time, because when you have more than 4 or 5 channels, then you cannot really do that anymore.
So I guess I'll launch another question if there's no one jumping in here. Back to the question of platforms. And so you mentioned, it seems like a number of your tools there have components that might be shared across the pipeline. Like maybe OpenSeaDragon for your, multi-scale, 2-dimensional sort of Google Map kind of approach to things. And I think you mentioned that you layer in some customized WebGL for like your lensing techniques and some of the other things that you're doing, like when you were showing the ability to rapidly show a lens and then the corresponding information. So is that all one common JavaScript base that you kind of build these different applications out of?
Yeah, that's a good question. I think it is. So we have a kind of a core functionality that is the multi-channel rendering, the cell-based segmentation rendering, the linkage to feature data, and the querying of the backend to get the respective tile and feature information. Also kind of, parts of the interface and the window management that you see there, and that basically we reuse for all these tools. Now, would it be great to have all the functionality in one tool? Probably for a user perspective. In terms of our research, we basically focus on a specific aspect, right? So yeah, it would be great, but it also, it would be a lot of integrational work, and I hope we can get there at some point. So some of these tools are more in use already. And some others are more research prototypes that are not yet, hosted somewhere where people can access them.
Yeah, I mean, a lot of the techniques that you showed were quite impressive, and I could certainly see them working in a three-dimensional space, right? Like maybe built on top of another tool like a neuroglancer style.
Yeah, that is
approach.
actually a very interesting point. We have tried out neuroglancer recently as part of a hackathon challenge that we designed for some colleagues, and they were able to load this multiplexed imaging data into neuroglancer. Use the different, segmentation layers as means for the image channels, and do some surface rendering on that in neuroglancer. I think volume rendering would be great in neuroglancer. We haven't really tried out that. I know is that in I think in an experimental stage there, but it could be interesting to look into.
Okay. Cool. Well, if there are no more questions, I think that thank you very much for your talk. It was great.
Well, I thank you very much for having me. It's an honor to talk here.
Oh, great. And, I'm sure that a number of people wanted to actually listen to this talk, but we'll probably get an opportunity once we post it onto YouTube, because a lot was covered here, and probably it's good to go back and take a look at some of these demos again. Yeah. Thank you.
Thank you.